experiments here. So I have these two samples, pork suet infused venison V3 and 2016 smoked ham V3, and I believe that I have successfully isolated a single species of bacteria. And I will explain in depth what species in this video. Okay, so first things first, we have to look at where I chose to isolate them from. I chose to isolate these samples from raw pork products. So I have found out that 35 to 70% of samples of raw pork products contain yersinia enterocolitica bacterium. So you have to look at the fact that this species of bacterium grows well, surprisingly well at refrigerator temperature, but also grows surprisingly well at room temperature. According to this article, yersinia enterocolitica can propagate and breed at refrigeration temperatures, but it is also a good breeder at temperatures up to 37 degrees Celsius, which is human body temperature. The coffee agar culture is a low pH and acidic, and yersinia enterocolitica, being a gastrointestinal parasite, bacteria, survives at surprisingly low pHs and acidic conditions. Also, the fact that the bacteria is a white to off-white color is also characteristic of this species. The shape of the bacteria colonies are also pretty consistent with my results. I'm not 100% sure about anything, but I am pretty sure that I did isolate this species. We have CIN agar for the selectional differentiation of yersinia enterocolitica. Anyway, here we go. I also have these three samples of bacterium: dog culture V6, Remnar gut flora, my gut flora, that I would very much so like to test. This is the pork suet infused venison. Unfortunately, however, my 2016 smoked ham culture got all moldy in the time waiting for this to get here, so I could only really use this one, unfortunately. Dog culture V6. To cool the inoculating loop, you place it on a sterile part of the petri dish until it cools. And I'm going to put that one on top of there. The next one is going to be what I believe to be Remnar, my cat. Oftentimes, animals are carriers of this bacteria, or even humans, which is why I tested myself and some of my animals. That's how CIN agar works. Okay, so it is a regular nutrient-rich agar, except for a couple things. First, it only has one carbohydrate, and that is mannitol. Mannitol is a type of sugar that is given to diabetic people, and very few animals can properly ferment it. However, Y. enterocolitica ferments it into an acid, and this causes the second thing. This culture has a pH indicator that turns pink when exposed to the acidic conditions, meaning that this means that the bacteria colonies on the CIN agar, if they are yersinia, or they will appear as a very distinctive white colony with a red bullseye because of the dyes that change color when the acid content goes up. So no other natural bacteria colonies look like a bullseye, because most of them are a uniform color, meaning that it is very easy to distinguish between Y. enterocolitica colonies on this agar. And if it is contaminated with other bacteria, the colonies will appear as like a less bullseye red. For the dish is upside down, Store for 48 hours at room temp or 24 hours in an incubator. Okay, so let's check out the cultures. It's been about 24 hours. Okay, so this is Remnar. There are a few dots of bacterial growth. They're a little bit red. However, I'm really not sure if that looks like Y enterocolitica. Okay, so luckily through all of my experiments, I didn't infect myself at all, since I see no bacterial growth on my culture, really. Dog culture. Most of the bacterial growth is relatively colorless, meaning that I'm guessing that my dog isn't infected. But pork suet infused venison. Look at that. Look at how much yersinia enterocolitica there is. That is insane. Wow. I mean, that is definitely Y enterocolitica. That, or at the very least, it is relatively impure Y enterocolitica. At the end there, you can almost see singular colonies. Okay, so I'm going to give these ones 24 more hours to incubate. And this one, I'm going to transfer right now. I have a brand new culture and a sterile an inoculating loop and a sterilizing lighter. And I believe it is time to choose which colony to take. I think I'm gonna take one of these red dots near the very end, since evidently they are definitely Y enterocolitica. Also, I made a mistake when streaking the culture last time. It was the first culture that I streaked, so I didn't really know how to do it properly. This new culture is nice and cold because I'm keeping it in my new mini fridge. The mini fridge also acts as a heater in an incubator, which I'm gonna eventually use it as. I heated this up with the lighter, and you put it on a sterile part of the new petri dish to cool it so that it doesn't kill the bacteria instantly when it touches the bacteria. As you can see, I think it's cool enough now. But I take one of these on the end, the smallest dot. Be very careful. And now I'm going to streak this area of the petri dish right here. Now, to spread out the bacteria and get individual colonies, I'm going to re-sterilize. And yeah, once you have recooled it, rub it into the already infected area to pick up individual bacteria, and then rub it in the next section of the petri dish. And you can repeat the process multiple times for better results. I suggest around 3 times. 4 times maximum, I believe. One more dilution. I'm just trying to get an individual single colony of Y enterocolitica, so that it is very pure, uncontaminated by other bacteria, because the pink color in this bacteria is strong. I bet it could be stronger. Get back on the bottom of the stack. I'm going to check on these in another 24 hours and this in another 48. Enjoyed, and see you in the next video. And until then, I'm probably going to use the rest of my CIN agar cultures. I'm trying to get a pure colony of this bacteria.